Right, thanks everyone. I'm going to speak about how large-scale brain network topology relates to the transcriptome and what that can tell us about how the brain is organized. No. Okay, I'll use this. Okay, so modern neuroscience is a very different landscape to traditional neuroscience. We have a lot of open data that's probing the brain in unprecedented detail, down to the cellular level in the case of some experiments, especially in mouse. And so what can we do with all this data? We're inundated by this unprecedented detail of data. How do we make sense of it? How do we understand how brains are organized? And how do we connect understanding of individual data types to each other? So I propose that three main promises of what we can do with this new data. So traditional labs might focus their expertise quite narrowly. The ability to now work across many open data sets allows us to integrate the expertise of individual labs and try and understand a bigger picture and how multimodal organization may manifest in the brain. And when we have an idea of a pattern or some constraint on brain organization in one species, the new open data sets also transcend species and make it very easy for us to bridge scales, looking at individual neuronal circuits and species, looking at from mouse all the way through to primate and human, and seeing what patterns match across scales and species, which then become strong candidates for telling us something that the brain might be valuing as something to be conserved. And then to kind of capture all these different individual insights, we need new theories that kind of make a coherent picture of the results of all these different individual experiments. So taking this kind of overview of what modern neuroscience data allows, I'll talk about two different themes in detail. The first will be some of our work on how we've connected data of different types and across different species. And the second, I'll present some recent work on the pitfalls you can get from applying conventional methods to these new data types and not thinking sufficiently about some of the subtleties which can manifest. So the first example I'll give is of hierarchical functional specialization. So we see lots of brain areas differ remarkably in their microstructural properties, their functional properties, their time scales. So you have all these interareal differences of lots of different types of properties. How can we make sense of that variation? Are they all varying independently? Do they all mainly vary along a common axis? And recently we showed that indeed they vary together along a common hierarchical axis. So here we show T1, T2 is a non-invasive MRI measure of hierarchical level in monkey. So we have the ground truth hierarchical level shown there to the right. And we show that in human, gene expression follows that T1, T2 map. So it's showing that microstructural properties are varying along this hierarchical axis in human. And this starts to give us an insight for what microcircuit properties underlie the hierarchical functional specialization. So the kind of fast, hide-wired dynamics of sensory areas differ remarkably from the slower integrative function of higher association areas. This might be underpinned by a particular microstructural variation characterized by these synaptic differences, cell type differences that vary continuously along this hierarchical axis. And so this was work in human and primate. The question is, does a similar pattern exist also in mouse? Previous work had shown that mouse cortical areas are in comparison very homogeneous in their properties. They don't vary so much. So how can we compare these findings in primate down to what we might expect in mouse? And so through this T1, T2 map, which you can measure non-invasively in both species, allowed us a bridge between results that had been established in primate and human all the way down to our new analysis, which added the mouse component. And so what we did was, for each gene, here every point in the plot indicates an individual gene, and we looked at its expression pattern across the cortex and computed the correlation between that expression pattern and the T1, T2 map. So in the top right there you can see p-valb. The horizontal axis is about 0.6, indicates that the expression of p-valb correlates at about 0.6 with the T1, T2 map in mouse. And when we do the same computation in human, this gives us our vertical axis. We see a very similar relationship in human. So for the interneuron marker, p-valb, we see that it's varying hierarchically in the same way in human and mouse. And this interspecies comparison is possible through this data-driven T1, T2 map. It doesn't require us to define a parcellation or a homology between cortical areas. And we see similar consistency with particularly NMDA and glutamate receptor genes. Another study we've done is looking at how structural connectivity properties relate to the properties of bold dynamics. So to do this, we, for each brain region, computed lots of properties of its bold dynamics, and we also computed lots of properties of its structural connectivity. And so this comparison between species doesn't require us to define a common spatial map, because we're analysing a correlation between two properties within a species, and we ask to see if that's similar across species. So when we look in mouse, we see that the high frequency power decreases within degree. So these brain regions with high degree have much less high frequency power. And when we run the same analysis in human, we see the same result. So this is telling us that brain regions that are strongly connected structurally have the slowest timescales of intrinsic dynamics. So these were also done with open data, and this allows us to connect common principles that are seen across scales and across species. Okay, I might skip that. And I'll skip. Arena is going to tell you the third example of this. This is to keep you around for the next talk. And in the remaining time, I'll talk about a kind of cautionary tale of how you can apply enrichment analyses to gene expression atlases. So I've seen a lot of examples of this, and our work has done this in the past as well. And so I'll talk through the subtleties of how it's done and what you have to look out for. So this is the situation, right? You have a phenotype you're interested in. It's a spatial map across brain areas. And you want to say, well, what types of genes are varying or are highly correlated with that phenotype? And if I see a particular class of genes that show a strong correlation to my phenotype, I might then interpret, well, those genes might have a function related to my phenotype of interest. So this is kind of how it's done. You have your phenotype represented as a vector across brain areas. You have a whole genome expression data where each gene has a particular transcriptional pattern across brain areas. And you can then score each gene according to how well correlated it is to a given gene's expression pattern. So each gene gets a score. Some genes are very highly correlated to my phenotype, some less so. And then we group them into an ontology of gene categories to say, well, which categories are concentrated in genes with high scores or genes that are strongly correlated to my phenotype of interest. And so this is very common, as I mentioned. It's been done in mouse with relation to network topology properties. People have tried to predict functional connectivity from gene expression. People have looked at how genes relate to intermodular hubs and found a metabolic connection. Some of our work looked at transcriptional patterns of rich club connectivity, and it's been done also in clinical groups and looking at the dynamic properties of spatiotemporal brain dynamics. And so what we noticed from all these different literature studies was that a common enrichment signature emerged. So a lot of papers shown here all flag as significant a similar Go category. So in the case of chemical synaptic transmission, despite working across different species, different phenotypes, different specific analyses and processing pipelines, a lot of people keep flagging the same types of Go categories, and often they're related to metabolism, behavior, and synaptic or neuronal function, including our own work. And when we surveyed the literature and gave each study that reports an enrichment a barcode across Go categories, so here we put a little blue dot if a particular study, which is a row in this matrix, found a particular Go category as significant, we can see a common structure emerging with lots of different studies keep flagging similar types of Go ontology terms. So it could be a coincidence that such diverse studies are getting similar patterns, or it could be a sign of methodological bias. So to kind of do a simple test for the possibility of bias, we generated lots of random phenotypes and repeated the analysis. So do some categories of genes show a stronger correlation to a completely random map with no structure? So our benchmark null was that we should get false positive significance at about 0.04%, or 0.4% rather. But when we used the real gene expression data, we found some Go categories being flagged as significant as much as 17% of the time in mouse, and up to 23% of the time in human. This indicates that just putting a purely random spatial map and computing enrichment under the conventional pipeline, 23% some Go categories are being flagged as significant to this random map. And the types of categories that show the strongest bias in this case were similar to those that have been flagged most in the literature. In this case, synaptic transmission and membrane potential, and in human, the false positive enrichment signal was biased towards metabolic Go categories. Now, given that gene expression is spatially correlated and most phenotypes in the brain are also spatially autocorrelated, this means that there's a physical effect. So nearby areas have a greater probability of being connected structurally, having more similar cytoarchitectural properties, and having similar gene expression. So this is another driver for the bias. And we showed that by, instead of generating random maps, now we generate maps with a spatial correlation that matches the types of phenotypes we see used in these analyses. So you can see there in the top right, you have this gradient-like pattern where nearby brain areas are more likely to have a similar phenotype map. And so when we quantify the bias against random maps and then against these spatially correlated maps, we see another increase. And the categories that are most affected are again those that are commonly reported in the literature for real phenotypes. And in the interest of time, I might skip towards the case study, where we tried to run this analysis in a real-world example. So in this case, we computed the structural connectivity degree of each brain area, and we asked the question, well, which genes are correlated to that degree? Now, when we use our conventional null model, where we just randomize genes, we find 600 significant categories. When we do our enrichment relative to random maps, we find 1,000 significant categories, and these categories are related to sensible metabolic and neuronal function. But interestingly, when we generated random spatially correlated maps and looked for Go categories that had a stronger enrichment signature to connectivity degree than expected from a random spatial lag model, we found no significance. So the spatial map of connectivity degree, despite being highly significant according to the conventional enrichment analysis, is actually consistent with random spatially correlated maps. This kind of framework gives us a way to test more and more subtle hypotheses through formulating different null models in a more subtle way than just applying conventional enrichment analysis. And when we digged into why that might be the case, we found it was driven by cortical and non-cortical expression. So connectivity degree is higher in the cortex, and most genes show a strong differentiation with cortical expression. Okay, so the interpretation of gene ontology enrichments should be revisited because of this strong bias. But most results in this area are unaffected. The correlational gene expression, specific hypothesis-driven analyses, and specific transcriptional trends all hold. Our analysis just shows a bias in applying conventional gene ontology enrichment to spatially embedded brain phenotypes. And this could drive the bias towards categories related to metabolism, synaptic function, etc. Okay, as I've said here. And some of our work is implicated. And in the interest of time, I'll skip through to thank collaborators, everyone who's made their data open, and I've given some references there. Thanks for your attention.